So our next speaker, our next chemistry laureate for 2014 is W.E. Moerner. He was born in 1953 in Pleasanton, California. He got his PhD in 1982 from Cornell University, and he is now the Harry S. Mosher Professor in Chemistry and Professor by courtesy of Applied Physics at Stanford, California. Welcome on stage.
Thank you. Well, thank you very much. I want to begin by thanking the Nobel Committee for Chemistry and the Royal Swedish Academy of Sciences and the Nobel Foundation for selecting me for this prize. I am truly honored to share the prize with my esteemed colleagues, Stefan Hell and Eric Betzig. For the inspirations for single molecules, which I want to talk about now, and why imaging and photocontrol and so forth become foundations for a different way of super resolution than what Professor Hell talked about, we have to go back to the mid-1980s. When I was inspired by the work of David Wineland and other people to look at single ions in traps in a vacuum, they were able to observe these single ions and do spectroscopy. But why not molecules? Why couldn't you observe a molecule with visible light, a single molecule? Well, of course, there's a bit of a problem, because this famous physicist who built the foundations of quantum mechanics, Erwin Schrödinger, felt that you could never experiment with just one electron or atom or molecule. And he wasn't the only one that thought a single molecule could not be detected. So a key aspect of the early days was getting to the point where we realized that we were able to do it, that it would be possible. And in order to explain that for this broad audience, I need to explain a bit more about spectroscopy. So let's go back to a simple experiment. Let's take this molecule, pteroline, and put it in a transparent host of paraterphenyl, and I'm showing its optical absorption as a function of wavelength. And here's the visible spectrum to sort of calibrate you as to what colors correspond to what wavelength. And if you imagine what's going on, at long wavelengths, there's low energy, and so you don't have any transitions. You go to shorter wavelengths, you begin to pump transitions on the molecule. Let's always focus on this lowest transition, or the first one that you come to. If wavelength increases to the right, then because frequency and wavelength are inversely related, then frequency would increase to the left. And the frequency here is about 460 terahertz, 460 times 10 to the 12th cycles per second. A very, very high frequency. 860, even higher over here. Well, now we're interested in doing low temperature experiments because that's where we first observed single molecules. So let's now expand this scale dramatically by a factor of 25 and cool to very low temperatures in liquid helium and see what happens. So here's what you see, and now I've reversed the direction of the scale. Now frequency is increasing to the right and wavelength increasing to the left. And note what happened to our color bar. We're only seeing a tiny part of orange here. You can see a slight change in color, but we've expanded it dramatically. Still, these represent different colors of light pumping the molecules, and what you see is very, very narrow transitions. The molecule absorptions became very narrow. In fact, you can see that there's 4 different origin sites, 4 different inequivalent sites for this molecule in this crystal. Now, what happens if you go even further down? Notice it's only a small range, only 11 terahertz range now being displayed. Let's now blow up the scale even more for this first transition, the lowest transition, by another factor of 1,000. What happens is that the molecule's absorptions get extremely, extremely narrow. Now I have a sort of a sketch of what's going on over here. The individual molecular absorptions are very, very small, and you don't see just one line. You see a profile that corresponds to different molecules which are in different environments, which have slightly different frequencies. We call this inhomogeneous broadening, and when I was at IBM Research in 1985, we were interested in studying this effect for a number of interesting reasons. You'd like to measure these widths of these individual molecules, and you have to use some tricks. For example, you could use photon echoes as one idea, but we were using spectral hole burning, because in the 70s, Dietrich Haar, Anshel Goroskovsky, and others showed that you could do something called hole burning. Now, what's that? Think of this inhomogeneously broadened line, and now if I irradiate it with a single frequency laser, which is very narrow, I can select just a small group of the molecules, and if I can change them somehow so that they move somewhere else in frequency space, I leave behind a dip, a lowered absorption, which was called a spectral hole. And the idea at IBM then was to use this spectral hole burning idea to achieve optical storage. Optical storage in the frequency domain. You could move to one frequency or another, and at each frequency you could choose 0 or 1. Do I write a hole or not write a hole? And that would be a way of storing digital information in these molecules. Much work was done to develop molecules to do this and methods to do this. But an exciting aspect for me at that time is that at IBM, not only was technology important, but science was also important. And I could ask this fundamental question: what are the ultimate limits to frequency domain optical storage if you use this hole burning idea? In particular, there's a question that came to our minds: is there a spectral noise of some sort that results from statistical number fluctuations or the discreteness of the individual molecules, which would define the smallest possible hole that could be written? Now, let me explain this with a simple analogy. Suppose you now have just 10 boxes and you've got 50 balls, and you want to throw these at these 10 boxes, with all boxes being equally likely. Well, then you might guess, one possibility is that you have exactly 5 molecules in every one of the boxes. But of course, you know that's not what really happens in a real experiment. In fact, what you really get are different numbers of molecules in the different boxes. In other words, the average might be 5, but in each box you get numbers that fluctuate about this average. And so this is the well-known number fluctuation effect that scales as the square root of n. In other words, in this case, the square root of 5 controls what's the variation that you get from box to box that arises from sampling. Well, now think of this as a frequency axis, just like I showed in that inhomogeneously broadened line, and make the inhomogeneously broadened line be extremely broad. And every one of these bins is a bin of width, the homogeneous width of the molecules. So that this basically means, this effect means that there should be some spectral roughness or fine structure on that inhomogeneously broadened line coming from the discreteness of the individual molecules. Here's a sort of a simulation to show you the same idea. At low numbers of molecules, you see a lot of roughness. The roughness seems to go down in relative size as you go to higher and higher concentration. And so this roughness is something that's coming from the discreteness of the number of molecules. But in the mid-1980s, this had not been observed. That's the key point here now. So we decided that we should try to observe this effect. So with Tom Carter, my postdoc, we first found the right material to do it. Pentacene molecules doped into a paraterphenyl crystal. And here's what the data look like. Here's a piece of the inhomogeneously broadened line, and you can see this amazing fine structure. Here's a blown up part of it where I show one measurement and then another measurement later, sometime later, and you see exactly the same structure. This is not noise. It's not something that's changing with time here, but it's a spectral roughness. Now, why is this interesting for single molecules? Well, it turns out that the absorption that you get, let's say for n molecules, scales linearly with the number of molecules, but these fluctuations have an amplitude that scale as the square root of n, as I've described. That's why we call it statistical fine structure. And this detection was achieved with FM spectroscopy, because FM spectroscopy only measures the difference of the absorption from the average value. It directly arises from the discreteness. And in fact, this is what gave us the feeling that a single molecule limit is within reach. In other words, if this absorption is coming from, let's say, 1,000 molecules that are absorbing at each wavelength, then these fluctuations are about the square root of 1,000, or about 32. This signal represents the signal from 32 molecules or so. And that means that our sensitivity of our apparatus only has to be increased by a factor of 32 to get to the single molecule limit. Therefore, it was going to be possible. So that's why Lothar Kador and I used this FM spectroscopy method from Gary Bjorklund to push that method to the single molecule limit, detecting single molecule absorption on the transmission of the light through the sample. Still using pentacene and paraterphenyl. A single molecule absorption looks like a single object there. FM makes a copy of it in 2 places, and another secondary modulation that we used takes a derivative of this, so the single molecule spectrum should look like a little W, and here you can see it in our samples. Here's no molecules and many molecules. Now, this was, of course, the hard way to do this experiment, but nevertheless, it had some interesting properties. It's insensitive to scattering. It is limited by laser shot noise. And of course, the challenge was, they made it hard, is that the focused laser intensity had to be kept very low, because if you broaden the molecules, then there's amplitude of these effects gets smaller and smaller. But at least it proved that a single molecule could be observed with visible light. The FM technique is one in which you take the laser and frequency modulate it. So here's frequency modulated light, here's the sample, and the sample converts this constant amplitude beam into an amplitude modulated beam. That's what you end up detecting. It's very much like FM radio at 506 terahertz, because that's the center frequency now of this huge inhomogeneously broadened line. A year later, a truly important advance occurred because Michel Laureate showed that you could also detect single molecule absorption using the emitted fluorescence. Here's some of his data. Here's an inhomogeneously broadened line, and these little dots are the individual molecules. This is the same system, inosine and paraterphenyl, but now you have a method that is sensitive to scattering from the sample. So these samples had to be incredibly clear and perfect, crystal clear, sublimed flakes so that you don't produce scattering backgrounds. Any sort of scattering background can make it harder to see the molecules, but that's possible to overcome. And so these methods produced higher signal-to-noise ratio for similar bandwidths. So essentially everyone switched to this fluorescence method after that. Well, here's some examples of single molecules at low temperatures from Pat Ambrose's work in my lab. Here's the inhomogeneously broadened line, and if you blow it up, you see these beautiful spikes, and in looking at just one, you see a width of about 7.6 megahertz in the visible, where the center of this is, as I said, 506 terahertz. That's an incredible resolution, spectral resolution of a factor of about 50 million improvement. That's why at this time we weren't really thinking a lot about spatial resolution, because we had such beautiful spectral resolution and could explore many properties. Nevertheless, we did take some images of these single molecules. And here's one where this is the frequency axis where we're tuning the laser, and this is a spatial axis where we're moving the laser spot over the molecule. And you can see they make little mountains in this three-dimensional representation. And we knew at that time that the width of the peaks along this position axis was basically a direct measure of the intensity profile of the laser beam inside the sample. In other words, you take this nanometer-sized molecule and move it over the laser beam spot, and you can measure its shape. That's very similar to what gets done for super resolution I'll talk about in a few moments. Also in those years, in the early 90s, in Urs Wildt's lab, a wide field two-dimensional microscopy was done and to image these single molecules at low temperatures. Well, if there were just images and just molecules that sat there or whatever, of course it's exciting to get to this new regime, to see single molecules. But what happened, which was even more exciting, is that surprises began to appear. Here I'm going to show some images, some spectra where the laser is scanning over 400 megahertz, looking at that single molecule, and look what happened. The molecule is jumping around. It's moving from one color to another. That is, even at liquid helium temperatures in a crystal, the molecule is jiggling and jumping from one frequency to another. It was amazing and really exciting to see this when Pat brought the data in and told me about this in the lab. And Jim Skinner helped with a theoretical understanding of this effect, that it's coming from fluctuations of two-level systems inside the crystal near the molecule. And when the nearby crystal changes, then the molecule moves to a different frequency. One of the cool surprises, that if you imagine putting a laser at a fixed frequency, then you'll see the molecule turning on and off. You'll see it blinking on and off as it moves into and out of resonance. Another key thing that occurred, Thomas Pache, another postdoc, looked at perilene in polyethylene, and he observed that you could optically change the positions of the molecules. Here's a molecule that's stable at that frequency, and then if you bring the laser into resonance with it, you can make it disappear. It's jumped somewhere else. You wait a little bit, and it's come back, and you can observe it again and again, and do this many, many times, on and off. So this is the early days, of course, of what you would call blinking and switching effects that end up to be useful later. Well, they're examples of why we were so excited about seeing single molecules in those days. We're in a complex system, and that's why removing the ensemble average is very important. You can then explore heterogeneity, see how the different molecules behave differently from one another. You can follow state changes in time and test theoretical effects. The molecule is a nanometer-sized object, so it senses nanometer-scale interactions, and it led to super-resolution imaging, as I'll describe in a moment. There's also commercial applications. Okay, so now let's jump to room temperature. I want to talk about a different thread of work that was going on during the 90s. Forgive me for this list, but I just want to mention some of the key advances that occurred. In the early 90s, 1990, several researchers looked at and saw signals from one fluorophore by measuring the autocorrelation signal from the sample, and single bursts were observed as well in 1990 for just a molecule in solution floating through a laser focus. Later, an important step was imaging a single fluorophore using near-field methods, which Eric Betzig achieved and we'll talk about. And then after that, several other groups also used near-field methods. Confocal imaging followed, then wide-field imaging followed, where you look at, in vitro, look at motors on an actin filament, Yanagita, and on the cell membrane, single molecules moving around, single lipids moving around. So there were a number of wonderful advances that caused us all to say, let's move to room temperature, where we can look at a lot of new biological effects and other chemical effects as well. So for all of those methods at room temperature, they roughly work like this for seeing a single molecule. You pump a transition from the ground state to the excited state and collect this emitted fluorescence. And so here's sort of a schematic of a sample where I'm showing that you use these kinds of molecules now, let's say rhodamines or other kinds of Psi3 dyes or even fluorescent proteins, and you often have them attached to the object of interest, and you want to be able to observe singles. Here's the focused laser spot, however, which is limited by diffraction to being only about λ/2 in width. And so that means that in order to make sure you look at only a single molecule, you have to dilute the molecules dramatically to have them be farther apart than the size of this effective focus spot to see single molecules. But in spite of that, much has been done and continues to be done with single molecules that are isolated. For example, I'll just show you very quickly a couple of them. Here is a cell, a CHO cell, and some transmembrane proteins have been labeled with a fluorophore. And if you watch this movie as a function of time, you see something really fantastic. The dance of molecules on the surface of that cell. This is what's going on in the cells in your body right now. The labels don't last forever, of course. You see them turning off, but the little tiny spots are telling us how the motion of these transmembrane proteins occurs on the surface. That's a diffusion effect that can be studied. Another kind of experiment can be done if you do time-lapse imaging. And here I'm showing the Colobacter crescentus bacterium, which is very small, only about a couple of microns long and 500 nanometers across. But Soyeon Kim observed a particular protein called MREB, labeled with yellow fluorescent protein. And what do you see on a time-lapse, long time scale? You see that this molecule is moving across the cell and then back around the backside. It's moving in a line. This is not at all diffusion like we saw before, but it's a completely different process, and this turns out to have something to do with the formation of the cell wall in these bacteria. Here are the tracks of many single molecules showing you how that occurs. And then the third example that I'm giving quickly is a material science example in a crystal at room temperature. Here you see two different kinds of molecules, some that are completely fixed, so the spots are not moving, and other molecules that are moving around like flies. They're actually doing that because they're moving in the cracks of the crystal. The moving molecules tell you where the defects are in the crystal. So you want to get the picture now that by watching these beautiful nanoscale light sources, they're very much like the stars in the night sky that tell us something, but here in living bacteria and in living cells, we can learn much more about the dynamics. Well, now let's go to the main thread of this whole Nobel Prize, which is super-resolution microscopy. And I want to summarize how you can use these single molecule labels to surpass the Abbe diffraction limit. The answer to that, and stated simply, is that you first have to be able to image these single molecules like I showed you, but then you have to add to that two key ideas. So the first key idea is called superlocalization. In the case of superlocalization, you can think about it in this way. Here is a little cinder cone mountain in the middle of Crater Lake in Oregon in the United States. And that scale bar there is 120 times 10 to the 9th nanometers. So this is human scale, of course. You know that if you take your cell phone, which has GPS capability, and walk to the top of this mountain, you can determine the position of the center of the mountain to very, very high precision. That's the basic idea for determining superlocalizing single molecules. Find the position by fitting the shape of the single molecule image. Illustrating that, here are some of these single molecule images, which are, they're all diffraction limited. That's why the blob has a size of about lambda/2. But if you blow up the image on your camera so that multiple pixels sample it, then this cross-section shows you that there's a shape to that blob. It's not just a disk. Some positions on the detector will count more photons, others less, and so that means that you can use a fitting function, in this case a Gaussian function, or more formally an Airy function, to fit that shape. The width of this is of course diffraction limited, but if the center position of it, the parameter of the fit that corresponds to where it's located, can be measured to much higher precision than the width. In fact, the estimates of the position of the center have this narrow green distribution with standard deviation sigma, and sigma is roughly given by the Abbe diffraction limit that we've been trying to beat, divided by the square root of the number of photons detected. As more and more photons are detected, you get better and better information about this shape so that you can find the position much better. For example, for 10 to the 2 photons, 100 photons, you can get down to 20 nanometer precision or so. Well, that works great, of course, for separated molecules and has been used a lot in biology. Here's a number of examples of very early superlocalization experiments where LDL particles or kinesin motors and so forth were imaged by finding a large object. But if you find the position of a single fluorophore, you can do something much better, because all the photons come from the same nanometer-sized object. And that means that you can then extract nanometer scale position information about where the molecule is. Watch this little demonstration in a simulation to see what I'm talking about here. Here's the image of the single molecule in time, and you see it's moving around, but each one of those shapes gets fit by a function, and therefore the error in the position is this green curve again. Here's the actual positions of the molecule as a function of time. We can measure these tracks beautifully now inside cells, for example. Well, the key point number 2 is that you want to go to super resolution. That is, you want to resolve 2 objects that are close together. And a key important paper was published in 1995 from Eric, where he suggested using an additional control variable to separate these diffraction-limited spots in the spatial dimension, and mentioned this spectral tunability that was available from the low-temperature single-molecule experiments. And that was actually then demonstrated by a group a couple of years later. A number of groups used several methods to try to separate molecules, either fluorescence lifetime or overlapping, or photo-bleaching, or differently colored probes and blinking and so forth. But these various early demonstrations only allowed you to separate a handful of molecules. What was needed was a real general method. And so that gives me key idea number 2. And I'd like to describe it in this general way by calling it active control of the emitter concentration and sequential imaging. So here's the idea. We want to observe this structure, and we decorate it with these labels that are fluorescent labels. Now the problem is, if you let all of them emit at the same time, then each one of those diffraction-limited spots will all overlap, and so you don't see the fine structure. So the solution is simply don't let them emit all at the same time. Use some sort of active mechanism that you have control over as the experimenter to keep the concentration low at any time. For example, let's suppose that we have a photoactivatable fluorophore that's dark all the time unless you turn it on with another control beam. Then you can do that at very low intensity and turn on only a few of them and localize those individuals. Then you photobleach and do it again. You'll get different samples of the underlying structure. And from these superlocalization positions that you record, then you put them all together in a pointillist-like reconstruction of the underlying structure. So that's the idea, breaking the diffraction limit using single molecules as tiny light sources. This idea appeared on the scene, and I heard it first from Eric Betzig in April 2006 when he described PALM at an NIH meeting in Washington. And quickly, STORM appears, FPALM appears, PAINT, DSTORM, all kinds of people showed that different mechanisms could be used for this active control idea. So it was a really incredibly exciting time to hear about all that. We did something called YFP reactivation then, and we didn't have an acronym for it, of course, so it was forgotten, right? So here's a new acronym I can add onto the pile that's mechanism independent, single molecule active control microscopy, or SMACM. So that's just for fun. I'm glad that you're laughing about it. Now, YFP reactivation, let's talk about that for a moment. Because when I heard about all these exciting developments, I said, well, how can we do this? What active control mechanism could we use? And I remembered this experiment that we did back in 1997. Rob Dixon was looking at and wanting with me to achieve the first imaging of a single fluorescent protein. We got some enhanced yellow fluorescent protein from Roger Chen, and here's one of the images that were recorded of the single molecules. But as before, just like at low temperature, when you first see a single molecule here at room temperature of a fluorescent protein, surprises might appear, and they definitely did. We saw that the fluorescent proteins were blinking. They would emit for a while and then turn off and turn back on again and turn off and turn back on again. This means that while we're getting a lot of photons in this emissive state, the protein goes into a dark state and turns off, and then thermally comes back and starts emitting again. Rob also saw that if he radiates the molecule for a long time, you go into a long-lived dark state, so it seems like it's completely photobleached. But if you use a little blue light, you can turn the fluorophore back on and observe it again for a long time until it finally goes into a long-lived dark state. A light-induced recovery of the molecules, another way to control the emission of single molecules that can be used for superresolution. These sorts of ideas, of course, spawned an industry of further development of switchable fluorescent proteins. Photoactive GFP, you'll probably hear about later. DRONPA and other sorts of fluorescent proteins were created where you try to optimize the switching mechanisms. What do the data look like from this blinking process in YFP? So here's some example. I show here some bacterial cells, and this is the white light image of the bacteria. And here's the fluorescence image, and now here's the movie. This is single YFP fused to a particular protein inside these bacterial cells, and you see all these little blinking stars that are coming from just inside each of these cells. That's so wonderfully amazing sort of kind of imaging that allows you to, in each frame of the movie, fit the positions of the molecules and then look at the structure of the underlying proteins in the bacteria. How about some additional examples? Let me show you. In a collaboration that's been going on in my lab with Lucy Shapiro for some years, we've been looking at Colobacter crescentus, an interesting bacterium. And here's 3 different proteins. Each case has been fused to YFP, MREB, PAR-A, and HU. These are the diffraction-limited images. And these are the ones that led people to say, oh, well, light microscopy won't be useful because it doesn't have enough resolution. But with these super resolution methods that I've described, this is what you see. This is, of course, the eureka moment that we've had when you are able to look inside a very, very tiny object and see far more detail and learn that even in these bacteria, there are structures that are helping to control the behavior and the development of this organism. This particular protein binds to DNA, so all of these bright spots are coming from the positions of the DNA inside the bacterial cell. So that's examples of using fluorescent proteins. What about using other methods of achieving super resolution? For example, that PAINT method. I'll describe it very quickly here. In this example, there is a cell off the screen, which is a PC12 cell, which is a neuronal-like cell, a model cell for neurons. And this is an axonal-like projection that comes out from the cell. And the way we're observing the system is to use this beautiful molecule that synthesized by people in Justin Dubois' lab, a molecule called saxitoxin, which is a toxin that binds to voltage-gated sodium channels. he can make it be fluorescent, so each of these spots is coming from this labeled saxitoxin as it binds to voltage-gated sodium channels in the membrane of this axonal projection. So remember, what's going on here is that in solution, these molecules are all floating around and they're dark because they're moving around. But when they bind to the surface, you see the bright spot clearly and can localize it and record it as part of the super-resolution image. The beauty of this particular experiment, since it's on a live cell, is that you can, as a function of time, observe how the cell changes. This particular display, I hope that you can see it clearly without too much washout by the lights, shows that each of these spots coming from voltage-gated sodium channels allows us to see how the neuron is changing with time. There are new neuritic projections that start appearing and disappearing during the time that this piece of the cell has been observed. The time resolution here is about 500 milliseconds. Okay. As one final example, I want to talk a little bit about the frontiers in this area. We have new fluorophore, we need new fluorophores, and we need new imaging methods. Let's have some quick examples of that. In the case of fluorophores, here's a particular interesting molecule called a rhodamine spiral lactam that has a closed form where the molecule is off and an open form where the molecule is fluorescent. And by modifying the structure of the molecule using friends in the synthetic chemistry from Bob Twiggs' lab, we can make this molecule switch with blue light so that you don't damage the cells as much. If you use this functionality to attach the molecule to the surface of the cell, this will bind to amines on the surface of a cell. Here are Caulobacter crescentus cells, where you can see the super-resolution reconstructions that show you clearly the subdiffraction stalks that are present on these cells that you don't see by normal microscopy. So that's just showing that there's great work to be done to develop, using chemistry, new molecules that can be used for these photo-switchable and photo-controllable mechanisms. In terms of methods, there's been a lot of interest in 3D methods. We've used this particular one where we convert the standard microscope into what we call a double helix microscope. The standard microscope, as you move in Z, you see a single molecule going in focus, out of focus, in focus, out of focus, and so forth. But with the double helix, the single molecule looks like two spots that revolve around one another. So I just, for a particular position Z, I just look at this angle of the two spots and I can tell you what Z is. All right? So you get X, Y, and Z from the camera, object depth being encoded into this revolution of the two spots. And this is what you see then for Caulobacter crescentus cells labeled on the surface with this molecule, but imaged in 3 dimensions using the double helix microscope. So what's so exciting about all of this work that's been going on is that it's had impact over many areas of science, in chemistry and physics and in biology. And this idea of working at the single molecule limit, this ultimate limit of individual emitters, has attracted the attention of many talented scientists who make lots of contributions. And I've listed more here, but there's no way I can possibly describe all the exciting things that are going on. And what's particularly exciting is that you can use these molecules now for going beyond the optical diffraction limit. So I have a lot of people to thank for making it possible for me to be here. This is thanks to the Moerner Lab alumni from IBM Almaden Research Center, from UC San Diego, and from Stanford University. But I also want to thank my current guacamole team. Here we are at Halloween, sort of dressed up a little bit in some funny way. We have this logo that says no ensemble averaging. Of course, what is this guacamole, right? What is that? Well, of course, one molecule is one guacamole. That is 1 over avocado's number of moles. So this is such a bad pun, I have to apologize to the memory of Amadeo Avogadro. I also want to thank my collaborators and colleagues at many locations, from WashU to Cornell to IBM to ETH, Zurich, UCSD, and Stanford. It's been truly wonderful working with all of them. I want to thank my mentors and the locations and the funding sources. Here's a couple of the key mentors. Here's Jim Miller from Washington University, my undergraduate institution, and Al Sievers from Cornell University, my graduate institution. But there were many professional mentors. I thank funding agencies who made a lot of this possible, and the support from all these institutions. And finally, I want to give thanks to my family and friends. Here's my stepmother, Esther Moerner, but here are my parents, who were the ones who taught me to love science and taught me to figure out how things work and how to fix things, which really made me an experimental scientist. And I, of course, owe everything to them. But also, I want to thank my son and my lovely wife, Sharon, who has been giving me a tremendous amount of support for more than 31 years. Thank you very much for your attention.